Hi there, everyone. Welcome to our second episode of the second season of our seminar series, The Future of Foraging. Very, very happy to welcome Dr. Holger Goerlitz and Abel Corver to this special episode on prey capture and hunting. We've got two really, really exciting talks coming up. I just introduced myself first. I'm Rory. I'm one of the co-organizers. And we also have Laura Grima and Dennis Goldschmidt as the other kind of co-organizers, and will be the panelists for today. So just to give a quick outline of how the structure of this will work, we're going to have two talks with 25 minutes each. Throughout the talks, feel free, audience, to write any questions that you might have for the speakers in the chat box or in the Ask a Question section. And as kind of moderator, I'll do my best to ask as many of them as possible to the speakers. So first we'll have a talk from Holger. Then we'll have, we're going to have 5 minutes of questions, roughly, and then we'll move on to Abel, who will do his talk. 5 minutes more of questions, and then the last sort of 30 minutes or so, we're looking to have a kind of more broad roundtable discussion on the kind of general theme of hunting and perhaps sensory ecology, etc. And again, you're very, very welcome to get involved with that either on the chat or the Ask a Question box. How about we kick it off? First of all, I'd like to introduce our first speaker, Dr. Holger Goerlitz. So Holger first entered academic science with a bachelor's in biology, graduating from Eberhard Karls Universität, Tübingen in 2004. And here he made his first contributions to acoustic ecology, publishing on the sensory ecology of lemurs. He then moved to Munich for his PhD at Ludwig Maximilians University, graduating in 2008. And here he worked on acoustic analyses of the hunting dynamics of a number of bat species. Then from 2008 to 2012, he did a postdoc at the University of Bristol, combining his previous knowledge on bat sensory ecology with an enhanced focus on the interacting sensory dynamics of their moth prey. And this work led him back to Munich, where in 2012 he joined the Max Planck Institute for Ornithology, which is now known as the Max Planck Institute for Biological Intelligence, where today he works as a group leader, based in part at the Savisen Research Station. His lab uses data-driven methods in the lab and field to study the acoustic and functional ecology governing the evolutionary arms race between bats and their prey. So without further ado, I welcome Dr. Holger Goerlitz. I'm just going to remove our co-host from the chat, and they'll be back in a second. And Holger, while we're doing that, feel free to share your screen.
All right. Thank you very much, Rory, for the introduction. On this screen, I think. Okay. Can you see the slides?
Yeah, that looks good.
Okay,
yes,
perfect.
We see everyone. I'm just going to mute myself.
Okay, thank you again, Rory, for the introduction, and thank you very much for inviting me. Hello, everybody, wherever you are, who you are. I think a large number of people there. So I'm going to talk a little bit today about what it is like to be a bat, and I'm trying to picture an active sound-based perception of the world. And you might notice I'm already playing a little bit with words here. But before we go into that, I don't want to forget all the people who actually doing all the work or did this work. So different members of my research group over the past years, amazing collaborators, lots of funding agencies that helped that. So thanks to all of them. And now let's go back to the work that all these people did. What it is like to be a bat. So this quote is from the philosopher Thomas Nagel from the 70s. It's quite famous in the bat world, but he was actually not interested in the biology of bats, but he used this phrase to think about the idea that it's actually very difficult to really understand how the world for somebody else is. It could be another human, you might perceive the world very different than I do. Probably you hear much better than I do. And then for animals, again, it's almost impossible for us to really understand how the world must be for them. Think about a dog who smells many things that we have no access to. Think about other physical stimuli out there, like magnetic fields that birds or turtles might use for migration. Even if going to vision that we rely on a lot, there are other animals that can see UV light, that can see polarization patterns, or mantis shrimps that have 12 different color channels. So their world is very different to ours. And this is well known, obviously, and already von Uexküll coined this phrase here, sensory Umwelt, so the sensory world of animals. Each animal has its own sensory world. From the large amount of physical stimuli that is out there, our sensory systems restrict ourselves to actually a very narrow amount of the information that the world is providing to us. And in my research, I'm focusing on acoustic information by looking at bats and their prey. And importantly, there are more than 1,400 different species of bats. So it's the second largest group of mammals. That means we cannot say that the bat perceives the world like this. But again, every bat species has a very different access to the world. And that's what we want to do today, have a short look at how bats perceive the world. And what you see here is a visualization of that from a recent movie about our work. A bat is flying through a forest. It's emitting echolocation calls. And the idea here is to visualize, to depict for us how the world for bat looks like. So the sound is traveling through the world. It's being reflected off this forest here and it's coming back. And there are a few things in here which are quite right. For example, the sound is traveling and it's reaching different trees at different times. But then there are many things that are completely wrong. For example, the spatial resolution that you can see here, individual leaves, tree trunks, that is completely wrong. A bat will never have such a high spatial resolution. And it will also represent the acoustic information as an image like we do. So again, it's very difficult to understand how the world looks like for other animals. Let's think about what an animal has to achieve. And in the auditory domain, this is called auditory scene analysis. We do the same thing. If you go into a new room or into a pub, you have to somehow arrange all the sounds and you depict internally an acoustic image of the world around you. So as the bat is flying around, it's emitting a call. It first of all has to detect a returning echo. So is there an echo, yes or no? It then has to separate this echo from background echoes, for example, from these leaves here. It has to localize it. So it has to measure the distance and the direction where the echo is coming from. Identification, is this an echo from an object that I'm interested in, for example, of this moth here? Or is it maybe actually an echo from a falling leaf? And the bat has to do noise rejection. So ignore all these other sounds, like echoes from ground, from trees, but also sounds from other bats or other insects out there, and also our human sounds that we are increasingly producing. Once the bat has an internal representation based on sound, it has to plan its behavior. For example, tracking this moth, or when another bat is coming, somehow interacting with that bat. So here is how such an interaction could look like. So there's a bat coming and another bat. They're aiming for something. Yes, there's a prey item. One of them gets lucky. The prey item didn't manage to escape. So these are typical interactions that we can observe in the wild. And this takes place in complete darkness, incredibly fast. This was a split second only. And somehow all these animals have to manage to get information, do not fly into each other, and navigate the space. So altogether, the research in my group is focused around these kind of aspects, and I want to organize this a little bit. So first of all, we're interested in how do animals perceive the world based on sound. Then next, prey is a specific part of the environment. So the next question is how do bats find their prey? And if you then look at predator-prey interaction, what does the prey do against that? So many moths, for example, have ears, and they react with evasive maneuvers. But there are actually many different bat species, as I said, and all of them have different calls, they live in different habitats, they have different food preferences. So this moth, with its very simple auditory system, has to correctly identify the danger by all those different bats and react appropriately. Likewise, we then have many different moth species with different traits, and they have to do the same thing. Furthermore, there are other animals out there that use sound for their own purpose. For example, bush crickets, for example, they sing to attract females. So this male here is singing to attract a female. At the same time, his song is audible to all those bats, and they use his song to catch him. So here is the trade of reproduction with predator avoidance. And lastly, all the echolocation calls by the bats are also audible by the other bats. So there's potential for communication within and across species boundaries, but also for interference between those species when they fly together. And lastly, all those biological processes are affected by external aspects, for example, environmental changes, variation, like weather conditions affect how sound is attenuated, but also our anthropogenic impacts like light or noise pollution. So what we have here is a very complex multi-species acoustic information network, which exists in pitch black darkness, and we try to disentangle that. So for today, I only want to focus on those aspects a little bit, look at the auditory strategies that bats are using to perceive the world, and a little bit then in the context of predator-prey interactions. Okay, let's look at bats. I want to start a little bit by showing you just a few exemplary echolocation calls, just to highlight how diverse they are. This is just a subset of a few European European species and echolocation calls. If you look worldwide, the diversity would be much larger. So you have time here. This is just 100 milliseconds, so relatively short. And here's frequency on the x-axis. Our human speech energies down here, our human hearing range up to 20 kilohertz at max. So bats are in the ultrasonic region. And here we have a mouse-eared bat, for example. The call is very short and covers a lot of different frequencies. So it sounds like chick, chick, chick. If you look at the pipistrelles, we see slight changes. There's some more energy concentrated in specific frequency bands. Nyctalus, again, very different, a much longer call with a lot of energy focused in a relatively small frequency range. The barber-style bat, we come to that later. It's a fascinating animal. Here it has multiple harmonics. So again, the frequency is differently distributed. And then horseshoe bats, rhinolophids, have a completely different call design. You see it's very long, 50, 80 milliseconds, and it sounds like a whistle. And all the frequencies in one particular frequency range. If you try to compare that to vision, then here we have one frequency, one wavelength, so just one light color, red, for example. Whereas here, we have many different frequencies, many wavelengths, so this is more like a colorful echo that the bats get back. And indeed, animals that, for example, try to find flowers, so they're bats that drink nectar from flowers, they have calls which allow them to distinguish a flower from a leaf based on frequency patterns. Whereas the horseshoe bats, they have a very different processing. In this case, because the call is so long, all the echoes will overlap in time. So they cannot temporally resolve that, but they're extremely sensitive to small little changes in frequency, and thereby they can perceive Doppler shifts, like shifts in frequency based on the beating wings of their prey. And this is like we know from some predators that hunt visually. If the animal is moving, they can perceive it. As soon as the animal is still, they don't really perceive it anymore. The same we actually have here. An animal which is not beating its wings is not perceptible by horseshoe bats. So we see a lot of diversity across species. If we now look within one species and actually within one individual, while it's doing different things, we can see again a lot of diversity. So here you see 1 second prior to an event, while the bat was either flying through an opening, drinking, landing, or hunting. And you see that the call patterning changes. In all cases, the bat is emitting more calls as it's getting to the object here. It's grouping the calls into temporal packages here. and the number of calls is increasing as the task complexity is also increasing. So if you just calculate the information update rate, the number of calls per second, then we see that here for the most complex task of hunting prey in the air, the bat is emitting up to 250 calls a second. So it's an extremely high information update rate, which it needs to constantly update the whereabouts of the insect and plan its behavior. So the bat has a lot of flexibility and active control over how it is sensing its environment. And it's a big advantage compared, again, to vision. We rely on the light that is there, but bats, by actively emitting energy into the environment, can control how much information and what kind of information they get back from the environment. And we can not only do this in the lab, as seen before, but nowadays with cool technology, they can put small little sensor tags with magnetometers, accelerometers, and microphones on the back of free-flying bats, and they really record what they're doing in their wild for a whole night. So here we have a mouse-eared bat at my field station in Bulgaria, and there's an animation of what it's doing. It's flying around, echolocating, and here catching a prey item. So here you see the echolocation sequence. This is the prey capture moment, and this corresponds to those colorful areas here. So these areas here, the blobs, they indicate the sensory volume, the space over which the bat, from where the bat can get an audible echo from a prey item. So usually the space is about 1 to 2 meters large, and this is the volume where the bat is searching for prey. Eventually, when it has detected something like here, it is reducing its calls. So it's producing more calls, as you've seen before, but it's reducing the amplitude. They become much fainter, and you see how the sensory volume is drastically shrinking. So the bat is actually focusing on the prey item, and by actively reducing the output volume, it is reducing its perceived sensory umwelt and thereby filtering out all background information. It's not getting any information from the ground, from the leaves anymore, and thereby simplifying the auditory scene that it's perceiving to completely focus here on the prey. We can also visualize this here. On the microphone, we can actually record the echoes that the bat is receiving. And here about again 1 second prior to prey capture, these bright dots here, these are the returning echoes, and we see they're getting closer and closer. This is the range to the prey, and the bat is closing in on the prey at constant speed and then catching it over here. So altogether, the bats use adaptive sensory sampling of the environment, and they adjust the sensing to the task, to the context, to the environment, to all kinds of parameters. And for us as researchers, this allows us to get very detailed insight into their sensory strategies. So now that we've seen how the bats adjust their echolocation behavior, we wanted to look a little bit what kind of information they can get from those echoes. So again, imagine a bat hunting for a prey item here on a water surface, which many species do because there are a lot of insects here. The bat is emitting a call and will get back echoes from this prey item. But at the same time, there might be more echoes, like from the leaves or even from the ripples or waves on the water surface. And we went to the lab and simulated this kind of situation by presenting the bat with these kind of rippled surfaces. And the bats were trained to discriminate a smooth from a rippled surface. And when they went to a smooth surface, they were rewarded. And these are all the different plates that Leonie presented. And we had plates with just a few ripples and many ripples, so from low spatial frequency to high spatial frequency, and with different height, from more than 30 millimeters, 3 centimeters, down to only 2 millimeters height. And what you see here, that is the performance of the bats, the sensitivity at which they were still able to discriminate the smooth from the rippled surface. So with increasing spatial frequency, the more ripples there were, the better was their performance. At the highest spatial frequency, they only needed a height of the ripples of about 3 millimeters. Well, it's not very large. So very tiny ripples are perceptible to them, and they can discriminate this from a smooth surface. Whereas if there are only a few ripples, even 32 millimeters were not enough to separate those two, the ripple from the smooth surface. So they're very sensitive to high spatial frequencies, and this is a typical high-pass filter. And interestingly, this is similar to our visual system. So these are the gray dots here. We as humans are also more sensitive to high-frequency spatial gratings of luminance patterns, so brightness patterns, these black and white stripes. So we're more sensitive to this one than to those low spatial frequencies. And we use this to separate foreground from background. High spatial frequencies are perceived as foreground and low spatial frequencies as background. And now we see the same thing here in bats, not for luminance patterns, but for distance patterns, these ripples here. But this must be with a completely different mechanism, because this perception, the visual system, this is a direct mechanism of our sensory epithelium. So think about your eye. Your eye has a two-dimensional retina, and a given light ray which is coming in is hitting a specific photoreceptor. And which photoreceptor is activated directly gives you information about the light direction where the light was coming from. In the auditory system, we have no spatial information at all from the beginning, because all the sounds, no matter where they're coming from, they all overlap at our eardrums, and all spatial information is completely lost. So the auditory brain has to completely compute spatial information. So the question is, where or how does the brain compute this spatial pattern here? So to analyze this, we set out to, we measured the echoes that are returning from those plates here, and then run them through a simulation of the auditory brainstem. So from the ear up to the auditory nerve that runs into the brain. So what you see here color-coded is the neural activity at the auditory brainstem as a function over time and frequency for all the 25 different ripples that we ripple plates that we had. Without going into detail, but just looking at the colors, you see there are some differences, there are probably also some similarities. And we can simply quantify the similarity with confusion matrix, and this is shown here. So yellow means high similarity, and here the plates now 1 to 8. These are here those on the left-hand side, the low spatial frequencies. They are all yellow, they're very similar. So there is no neural activity that can discriminate those plates. But for the rest of the plates, all those patterns are sufficiently different for the bat, for the auditory brain to discriminate between those echo patterns. So what we suggest is that the auditory processing exploits object-generated echo information to have access to the spatial pattern of the environment. The visual system does this by having a two-dimensional retina. So this can directly analyze luminance patterns by the activation pattern on the retina. The auditory system cannot do this and therefore relies on information that is directly contained in the echo. So we have convergent evolution here in the auditory and visual processing of space. Both systems have access to spatial patterns, but based on completely different mechanisms, depending on their sensory epithelium. And as in the visual system, we suggest that this is the basis for separating foreground from background. Like a prey item is producing high spatial frequencies, and the bats can use this to separate it from low spatial background frequencies. Okay, finally, let's move quickly to predator-prey interactions. Just to remind you again how important sensory systems are for predator-prey interactions. Like prey is looking out for predators, they might call and warn each other. And here we see different sensory strategies against prey, like warning colorations or mimicry or camouflage. And we know this mostly again from the visual domain and much less from the auditory domain. Okay, what do bats do when they catch prey? So one important thing to notice, bats are loud. They are flying jackhammers. Here are just different publications. They show that bats are somewhere between 120 to 130 decibels loud. So what does this mean? You might not know these numbers. So a jackhammer is at 130 decibels roughly, and also our pain threshold is in this order of magnitude. Whereas if we normally talk, our conversation is somewhere between 60 to 70 decibels. And this is a log scale, so this is extremely loud up here. And why are bats so loud? Because their calls are ultrasonic. That means they attenuate very quickly in the air. So if they want to detect objects, they need to be loud. And we can calculate the detection distances, and this is what we've done here. So to detect actually a relatively large prey item, even when they're so loud, they can maximally do this over 8, 9, 10 meters. So they're not very much looking ahead. And now the interesting aspect is moths, they don't have very sensitive hearing, but because the sound has to travel to them only one way, they can detect those loud bats actually at larger distances. So they hear the bat before the bat knows that there's a prey item and fly away. And indeed, if we look at the poo of those bats under the microscope, then we see very little remains of moths. Okay, now comes the barbicel bat that I mentioned initially. This species is extremely faint for that, 10 to 100 times fainter than all the other animals. And faint for bat means it's still as loud as a chainsaw. So notice what's happening here. If this bat is now closing on a prey item, it will detect this prey at about 2 meter distance when the prey is not yet aware of the attacking bat. And if we look at its diet or its poo, then we see with microscope or with modern molecular method that a very large proportion of the prey very large proportion of the diet consists of moths with ears actually. So the barbicel bat can hunt eared prey. And then we were thinking, if the barbicel bat, once it has detected the prey, reduces its call amplitude even further, as the other bats are also doing, it might remain undetected all the time. So Daniel went out to the field with a microphone array. He tracked the position of the bat. He put a moth up there with a tiny microphone just next to it. So we could record what the bat was doing and what the moth was hearing. And indeed, as the bat is approaching the moth at about 2 meter distance, 1 to 2 meter, it starts reducing its call level. And this is the level at the moth. Initially, the moth, as the bat is getting closer, is getting louder amplitude, but then the received level at the moth stays within this range here. And the lower red line, this is the neural hearing threshold of the moth. And the upper red line is the moth reaction threshold when they start escaping. So the bat manages to just stay below the moth reaction threshold, meaning that the moth will never fly away from the barbicel bat. So we see, as also before, low amplitude echolocation means a reduced and focused perception. The bat can only detect the prey at very short distances, but this prevents that the bat is detected by the prey, and the barbicel bat actually remains undetected by the moth until the final attack. And we sold this, was a nice paper, as a coevolutionary response of the barbicel bat to hearing in its prey. But then later we actually started thinking and realized that actually other bat species are also relatively faint. For example, this nectar drinking bat here. And they can also catch eared moths. But they are for sure not faint because they want to catch eared moths, but they are faint because they're flying next to this vegetation all the time, and they want to avoid getting very loud echoes from this stem here, from the leaves and so on. So we have an alternative hypothesis that hunting for eared prey in the open air is actually a secondary exploitation of a pre-adaptation to hunting close to vegetation. If we look at the phylogeny of the barbicel bat, then here we have the barbicel bat, which has faint calls and hunting aerial prey. Then we see that in this clade, we have many other species that we know that are faint or which are very likely faint. And some of them do aerial hawking, these are the blue dots, but most of them do so-called gleaning. So it means they take insect off surface structures. So we now think that there was not a coevolutionary response in the barbicel bat, but actually somewhere down here, the bats became quiet as an adaptation to flying very close to vegetation structures. And then secondarily, the barbicel bat use this again to hunt eared prey. Yeah, that's what I just said here. We have the secondary exploitation of an adaptation to reduce vegetation echoes. For the last 2 minutes, I now want to look at the prey side. As I mentioned initially, this male bush cricket here is singing, attracting females, but his song is also audible to the bat here. So he has to trade off reproduction, like attracting females with his song, and survival. He doesn't want to attract bats with that. What he can do is song cessation, like stopping to sing. And this is, if you again compare it to the visual world, the acoustic analog to hiding. Instead of running into burrow to be out of sight, he just has to stop singing, and then the bat has no cue anymore where he's sitting. So Tony went to the field, caught some of those bush crickets, brought them into the lab. The males were singing, and then she played different kind of calls to the male, and we analyzed for how long he was stopping his song. So here you see a control sound, a cricket, and a very high predation threat, which is a very loud bat call. And we present the stimuli for 1 second, and then you see the pause duration. He stopped singing in response to the cricket sound, but then before the cricket song actually stopped, he started singing again. His pause was only less than 500 milliseconds long. But for a very loud bat call, he made pauses between 2 to 5 milliseconds. So there is acoustic hiding as an anti-predator strategy. Next, we present a different kind of predation threat, so bat calls of different amplitude, and then we see with reducing predation threat, his pauses are getting shorter, so he's adjusting his response to the predation threat. And I think the very cool thing is we tested young and old males at the beginning and the end of the reproductive season. Old males, the gray areas, they make shorter pauses than the young ones. So the old males are taking a higher risk. They're more willing to becoming prey to a bat, most likely because they're at the end of their life anyway, and they are trying to get the last mating and therefore continue singing for longer to attract females. So with this, I briefly like to summarize. We looked at auditory scene analysis and behavioral strategies that bats use to simplify the auditory world. For example, they change call amplitude, they change the timing, the frequency of the calls. They also change the spatial pattern where they emit their calls. We have looked at that. And they're very loud. And if you compare this to visual systems, then we see convergent evolution of sensory strategies across sensory systems. So the environment or given tasks require an animal to get a specific kind of information. And independent of the sensory systems, animals are often able to get this information that they need. And this allows us to get very fine-scale experimental access to the sensory behavioral strategies. And lastly, briefly for the prey, they don't want to get eaten. They fight back. Despite, for example, those moths having very simple sensory systems with only 1 to 4 auditory neurons. And again, what I find really cool, we find analog anti-predator strategies in different sensory systems. So hiding, for example, but also many others. Okay, thank you very much for listening.
Brilliant. Thanks so much for that, Holger. That was absolutely fascinating. Yeah, as I mentioned to the audience before, if you have any questions, absolutely feel free to write them in the chat. I think we've just got one now, actually. So this one's from Laura. She asks, have you explored how bats respond when they detect multiple prey animals?
Very complicated topic. We don't, but there had been a very cool study from a Japanese group, and they tracked free-flying bats and free-flying bats that they're hunting for different insects, and they suggested that the bats were actually already planning their trajectory in such a way that they also, while catching the first insect, already in a good position to then intercept the next one. Right.
Okay, interesting. Yeah. I also have a question. You mentioned briefly about these kind of anthropogenic effects that that might impact the way that the bats forage. Is there much information on how that's affecting bats, or also whether they have is there any evidence of plasticity in the bats' strategies of hunting?
Yes, there
in
is.
reaction to
so
that?
The best understood strategy is the lumbar effect, which you might know or heard about. This is simply raising your voice, talking louder when there's a lot of background noise. So again, if you're in a pub or a concert, at the end you might have a hoarse voice because you talk simply louder, and you don't even really notice this. And across the animal kingdom, many animals do this. If there's more background noise, they become louder. And bats do the same, and this helps them to reject this noise. And this is particularly important for those bats that listen for prey-generated sounds, like this bush cricket song, or others, they listen for the rustling sounds of insects. Sorry, I'm mixing up things. The background noise is particularly problematic for those species that use prey-generated sounds. So if a beetle is walking on the ground and making noise, the bats cannot compensate this by raising their own voice. So a colleague of mine, they did tests and simulated traffic noise, for example, and many tens of meters on either side of roads, we are basically degrading the habitat for these kind of bats because the noise from passing cars makes it more difficult for bats to detect prey items. But it's the same for the echolocation calls, and we see that bats have strategies. For example, they might change frequencies, but the best understood strategy is raising the voice. And this can have potential costs, right? If you're anyway calling at the top of your voice already, and then you need to become even louder, that can have metabolic costs or straining your vocal folds, for example.
Yeah, very interesting. It'll be interesting to see how that develops over the coming years. Yeah. Maybe we have time for just one more question from the audience. So Marisol asks, she says, thanks for the talk, excellent knowledge. Would you please expand a little bit on the bat's brain adaptation to detect direction? I cannot catch completely the comparison.
I wanted to highlight the difference between the sensory systems before, somewhat early in the talk. So our retina is two-dimensional, right? So we have an array of many different photoreceptors, and light rays are coming in from different directions, and we get a real image on our retina. And this information is completely missing for the ear. And the auditory brainstem, or the auditory brain has extremely high computational power and is calculating direction. The same for us. And one of the key parameters, for example, is time differences. If a sound is coming from one direction, it will first arrive at this ear and somewhat later at the other ear. If the sound is coming directly from the front or from the behind, it will arrive at both ears at the same time. And the brain can analyze these kind of time differences to get the direction. Also, intensity differences, the sound coming from this side will be louder at this ear than at this ear, and the brain is analyzing that. And then from the sound that is coming from in front of me, but from above or directly in front of me, will have different frequency patterns based on my pinna. So here, your outer ears, they attenuate some of the frequencies and amplify others. And depending on the direction, this gives frequency information that the brain is also analyzing. And then for the bat, that's a special case because it knows when it has emitted a call, the time delay or the time that it takes for the echo to return gives its direct information how far away the sound is. That is something special that we don't have, right? Because if we hear a sound, we don't know when it started.
Yeah, fascinating. So yeah, thanks a lot. We actually still have some more questions in the chat. Perhaps during the talks, like if you get the chance, you could maybe write some messages. Otherwise, no worries. Maybe we can cover them in the discussion. But for now, I guess it's time to move on to Abel. I will.
I will take over now. Yeah. So welcome also from my side. It's really my honor to introduce our second speaker today. So we're now moving from the world of incredible bats to incredible spiders. And our second speaker today is Abel Corver, and he's a PhD student in Andrew Gorda's lab at Johns Hopkins University. And he actually obtained his bachelor's degree in linguistics, am I right, at Harvard University. Before then joining Vivek Jayaraman's lab at HHMI Geneva for 1 year. And then in 2017, he started his PhD in neuroscience at Johns Hopkins University. And Abel is interested in how neural circuits generally regulate complex and cognitive functions, such as in language and navigation. And today he's talking about his current work on studying decision-making in orb weaving spiders during web building. And yeah, with that, I will lead over to you, Abel, and I'm really excited to hear your talk.
Great, can everyone hear me? Perfect. Thank you so much, Dennis, for that kind introduction. And thanks for the talk, Holger. That was very, very interesting. I'm very excited for the sort of joint discussion afterwards. Let me share my slides. Okay, I think everyone can see the slides, but let me know if there is a technical issue. So thanks again for the invitation. I'm very excited to be able to share with you some of my ongoing work in Andrew Gordas' lab, trying to understand the webmaking behavior of this small webmaking spider, Euloborus diversus. Now, many of us have probably been struck by the beauty and symmetry of final orb webs. However, this symmetry conceals the multiple simultaneous physical purposes served by the web. Now, because orb webs are the only source of food for orb-weaving spiders, their primary purpose is, of course, to capture prey. But before prey can be captured, the web needs to absorb and dissipate the tremendous amount of kinetic energy impacted upon it by various sizes of prey. A current body of research has shown that different types of geometry in the web, such as the capture spiral versus radii, play different physical roles in dissipating such energy. However, once prey are captured, the specific structure of the web continues to play an important role in helping the spider to locate the prey by transducing not only the vibrations emanating from the prey itself, but also by transducing the active vibrations introduced by the spider itself. There's much interesting work on this topic. And in fact, a fellow graduate student in our lab, Shini Hung, is using high-speed videography combined with Fourier analysis to visualize the spatiotemporal pattern of these frequencies as they travel through the web during prey localization. Now, a unique property of webmaking is that in addition to having to satisfy these previous two physical constraints, the web is also the only substrate for movement for the spider itself, further constraining this behavior. So despite this emerging understanding of the multi-purpose physical properties of the web, it remains incompletely understood how the many thousands of moment-to-moment decisions on the part of the spider yield this ultimate structure. For example, the spider frequently executes silk pulling, as well as anchoring lines together and frequent leg probing. And during these motor actions, during web construction, the spider simultaneously receives a range of sensory input stemming from the web in the form of its tactile interactions. And because this particular spider tends to build in the dark overnight and its eyes are facing away from the web during web building, this is an exclusively tactile behavior. We can therefore think of understanding the webmaking algorithm in terms of understanding the real-time interaction between these sensory and motor factors. For example, the brain of the spider may encode preferential transitions between certain motor actions, and those motor transitions may be modulated by the real-time sensory feedback that the spider receives from the web. In fact, some in the field have postulated that webmaking could be almost exclusively explained by such reflexive sensory motor rules. However, the extraordinary robustness of this behavior to both sensory noise as well as ambiguity to me at least suggests that there may be a role for internal states in guiding the web making process, either in the form of a memory of the spider's position on the web, as well as perhaps a memory of the current stage of web building. Now, despite longstanding interest in the field in documenting these sensory motor patterns, a longstanding challenge has been documenting and analyzing these rapid transitions, these rapid sequences of motor actions and sensory inputs. And so today I'll share with you some of our past and ongoing work quantifying not only the motor sequences underlying each of the stages of construction, which will be the first part of the talk, but also our ongoing work attempting to understand the real-time sensory landscape during the construction of the web. Before we proceed with that, just a recap of why spiders are, I think, such an interesting model system for studying complex and cognitive behaviors. First of all, their behavior is not only highly complex, but the webs produced by many orb weavers, including this particular spider, has geometry that is sparse enough that it can be completely annotated and thus quantified geometrically. Furthermore, even though the spider looks very large on this slide, it's in fact only a couple of millimeters large with a brain about the size of Drosophila, thus potentially facilitating circuit mapping and physiology in the future. And most importantly, web making in all orb weavers proceeds along very well-characterized and distinct geometrical phases that collectively last many hours. And therefore, both the short and long timescales exhibited during web-making behaviors span the range of cognitive timescales exhibited by other animals, including ourselves. So what are these stages of construction I was referring to? When a spider first comes across a building site, it will build an exploratory structure called the protoweb, which you can see in blue. And once it is motivated to hunt at that location, it will remodel that structure into a radial structure, followed by a temporary spiral called the auxiliary spiral, which then helps the spider build the final capture spiral, which actually entangles prey. Now, in some individual instances, individual spiders may decide to add a dense patch of silk at the hub called the stabilimentum, which perhaps plays a role in acting as camouflage for the spider. So despite this longstanding interest in these stages of construction, a challenge has been documenting simultaneously both the motor output of the animal as well as the sensory input in the form of the geometry of the web. So to that end, we have developed the following behavioral assay in which the spider builds a web in a 10-centimeter diameter arena, while it is illuminated alternately by a top-down infrared light and a bottom-up oblique infrared light. And therefore, using just a single camera, we are essentially capturing two separate video feeds, one displaying the movements of the spider at 50 hertz, and one capturing the sensory landscape at 10 hertz. We can then align these, we can then interpolate these two representations to yield a simultaneous view of the spider's posture and tactile interactions with the evolving web geometry. Now, because we are interested in understanding motor patterns during web construction, we of course need to know the posture of the spider at every moment in time. So to do that, we trained a deep neural network for posture tracking called DeepLabCut based on 10,000 manually annotated examples. And you can see the automated predictions of this network superimposed on the video on this slide in the form of the red dots. And the confidence interval of that tracking is again shown in the bottom left, showing fairly accurate tracking over the course of this behavior. Now that we have access to the sort of continuous posture dynamics of the animal, we were interested in the question of if there is a sort of alphabet of stereotype motor actions underlying this behavior. So to that end, we would like to be able to compare the similarity and dissimilarity of different movements to find sort of stereotype motifs. However, a problem that arises in the case of spider motor behavior is that because the spider is only able to place its legs in those places on the web where there is existing web geometry, the heterogeneity of the silk geometry at any moment in time essentially induces variability in the details of the execution of the spider's movements. We therefore defined the following movement similarity metric, which we call the posture alignment metric, in which we take two 1-second clips, either from the same spider at different moments in time or from different spiders. And we both spatially and temporally try to align the time series of each limb coordinate and sum the remaining squared errors, yielding a single number representing either the similarity or dissimilarity of those two movements. Now, the space of all possible movements is, of course, inherently high-dimensional. So to discover the lower dimensional stereotyped structure of the spider's movement space, we use t-SNE dimensionality reduction combined with this custom movement metric to create maps of stereotyped movements separately for the anterior and posterior legs, which you can see on this slide. And the reason we do this separately for anterior and posterior legs is that in our experience, they can execute their actions quite independently. As you can see in this density, as you can see in these t-SNE maps, there appears to be a clustered structure in this t-SNE space. And so we use watershed segmentation followed by manual simplification to create a set of 9 stereotyped movement motifs that we observe during the web-making behavior, ranging from left and right leg sweeps to walking and silk anchoring behaviors, as well as 2 different types of silk manipulation. Now, with this movement classification in hand, we can apply this to the remainder of our recordings, which I've done here for an example capture spiral segment. So here you can see in green in the top center, frequent use of fast silk pulling, which is very characteristic of the capture spiral stage. And in the top left, you can see in orange the regular occurrence of anchoring behaviors, which occur every time a new segment of capture spiral is attached to an existing radii. As we saw before, orb weaving proceeds along geometrically distinct stages of construction. However, we wondered whether those geometrical stages in fact emerge from distinct sequencing of actions during these stages. So to answer that question, we constructed several hierarchical hidden Markov models in which the model was able to observe the transitions between motor actions at any given moment in time, but had to infer the hidden behavioral regime that is modulating the likelihood of transition between different motor actions. And in fact, when we fit those hierarchical hidden Markov models to our behavioral data sets, what we find here on the 3rd row and below is that it largely replicates the segmentation of the web-making behavior that we previously achieved manually based on the geometry of those stages, indicating that most of these stages appear to be characterized by distinct sequencing of actions. And in fact, when we apply the same hierarchical hidden Markov model across our 21 recordings in this first data set, what we find is that the model is largely consistent in how it segments each of the recordings, indicating that these distinct action sequences are largely stereotyped across individuals. Now we fit models with different numbers of hidden regimes. And of course, although the performance of these models will always increase for the in-sample data, we find that the predictive power of these models on fitting out of sample data reaches its peak around 5 or 6 regimes, which is approximately the same as the number of sort of geometrical regimes that we know about. You may have observed from the previous slide that even though the segmentation of the behavior based on the movement patterns largely recapitulates the segmentation based on geometry, there is one interesting caveat to this, which is that the hierarchical hidden Markov model does not clearly segment the protoweb and radii stages. And this is interesting because manually, it is often quite difficult to define a hard boundary between these 2 stages, and the transition from irregular protoweb to radii, despite the distinct names for those stages, in fact, is often much more blurry than the names suggest. However, at the end of the day, these structures are, of course, still similar. So what explains this discrepancy? Well, although this hierarchical hidden Markov model illustrates that at the level of Markovian movement transitions, these 2 behavioral regimes are highly similar. What it seems to suggest is that the where and when of where those movement transitions occur is different. And of course, that is fundamentally a result of the sensory input that may trigger these transitions during web building. As I illustrated before, our assay allows us to simultaneously record not only the motor output, but also the sensory input of the spider. However, even though we can annotate the geometry of this web quite accurately manually, this is quite prohibitive to do for the many millions of frames across our recording data sets. So we've therefore been developing an automated geometrical annotation pipeline that can do this for us for all our recordings. So after aligning the spider's body onto the web, we remove any parts from the image that are not easily approximated by line segments. We then compute a gradient map where the value of each pixel in this image represents the apparent orientation of the local geometry at that position. Now, one thing that would greatly help us here in tracking the geometry and to increase the signal-to-noise ratio would be to integrate the exposure of each image across frames. However, this is made impossible by the fact that the geometry is deformed by the spider's own movements as it traverses the web. We therefore use the following approach, in which we create several particle filters that are tuned to find points in the image representing intersections of 3 or more lines. And by constructing several thousand of these particle trackers, we are able to, over the course of this recording, track the deformation of the web during the spider's movement across it. This then significantly facilitates the next step of automatically detecting web geometry by allowing us to integrate and evaluate the connections of lines, not just for single frames, but across many tens or hundreds of frames, thus increasing both the accuracy as well as the temporal consistency of this inferred geometrical structure. And this is ongoing work to improve this pipeline. Now that we have reconstructed the geometry of the web, because we also have access to the posture of the spider, we can reconstruct the points of contact between the spider's body and the web. And that allows us to get back to our original question, which is, what are the sensory motor rules that may underlie web making? And in fact, if this behavior is indeed mostly reflexive, we should be able to formulate a relationship between this parameterized sensory input vector and some representation of the motor output. One way to think about that regression is in terms of mapping the silk lines that are converging on each of the spider's legs at any given moment in time, which I'm displaying here in the top left. And we can relate that sensory input vector to the subsequent motor behaviors that the spider executes at the next time step. And here the white ellipses represents a rolling window of the swept area of each of the legs, sort of indicating instances of sort of leg probing. And as we saw in some of the videos before, this active leg probing is a very important type of feedback that the spider receives during the web building behavior. Another way to think about motor output, though, is in terms of the silk changes that result from these leg movements. And so there is an interesting dual way in which we can think about this motor output, either in terms of the leg movements themselves or in terms of the resulting geometrical edits to the structure at any given moment in time. So here I'm plotting sort of time-lapse animation where the color scale represents this changing model of the web. And even though the spider is not visible, you can see new capture spiral lines being sort of put in their place. However, the creation of new lines is not the only type of motor output that the spider engages in. For example, especially during the proto-web stage, the spider engages in complicated rearrangements of tension and sort of the distribution of geometry, which you can see illustrated here. And what you can also see in the same animation is the collapsing of two what used to be separate lines onto a single line, essentially deleting geometry. So especially the proto-web stage, but also the other stages highlight this sort of intricate ability of the spider to edit the geometry of the web. And of course, this relates back to the earlier question of what are the sort of fitness functions that the spider is optimizing when it is executing these behavioral rules. So in our future work, we will aim to relate this representation of sensory input that I just showed you to these various ways of thinking about motor output. However, it is not clear whether such a reflexive regression relationship will be able to capture the full variability and be able to predict the full behavior of the spider. In fact, there may be a need for postulating internal state dynamics into these models. And by evaluating different types of such regressions, we hope to statistically be able to derive likely representations, likely sensory motor representations and internal states that the spider uses during this web-making behavior. Now, such models, being able to mechanistically extract potential internal state dynamics has interesting connections to a range of other experiments going on both in our own lab and in the field, and in the field as a whole. For example, it is known that the spider is able to repair web damage and resume after other types of interruptions. And interestingly, it is known that different types of drugs, such as ecdysone and LSD, have specific effects on particular aspects of the behavior. And so therefore, having an understanding of the internal state dynamics based on our observations of the behavior may be able to mechanistically explain the pathways by which such different drugs have their specific effects. And finally, in the long run, of course, we would like to use functional imaging in the spider to directly probe the dynamics of those internal states in the brain. So in the last part of my talk, I'd like to quickly summarize why we are so excited about the possibility of doing physiology in this spider going forward. Because despite longstanding interest in physiology in spiders, a continuing set of practical challenges have made this type of work more challenging. For example, many spider species have venom glands above the brain, making in vivo dissection significantly more difficult. And importantly, many species of spiders have very high internal body pressure, which they use to extend their limbs. And of course, that has made making imaging windows in these spiders significantly more complicated. However, this particular species of Euloborus diversus that we have started studying lacks venom glands and has fairly low internal body pressure because it spends most of its time hanging upside down from horizontal webs, therefore making in vivo imaging significantly more easy. And of course, its small brain should additionally help with imaging in this species. We've developed the following assay in which developed a custom 3D printed holder to match the sort of unique morphology of the spider's relatively large abdomen compared to its thorax, thus allowing, in theory, whole brain imaging. Our tethering protocol, we start by tethering the spider and we align it to this holder, after which we seal it to the well and we use an insect dissection pin to dissect an imaging window. And we are particularly interested in a structure called the arcuate body, which has been debated to be a potential site for tactile integration in this species, because, as I mentioned in the introduction, the ability of the spider to detect timing differences in vibration signals between its legs is particularly important when localizing prey on the web. But those experiments have so far mostly been done in individual legs. And we're excited to see how central brain structures, such as the arcuate body, may be playing a role in integrating these different tactile signals. So with that, I'd like to thank you all for your attention, and I'm very much looking forward to the discussion.
Many thanks for that, Abel. That was another very, very interesting talk. As I said previously, audience, if you have any questions, feel free to drop them in the chat. But while we wait, first of all, I will just re-invite the other panelists, etc., back onto screen. Let me just grab them. But yeah, that was fantastic. Thanks so much. One question I had was, is there much evidence that environmental effects, like idiosyncrasies within the environment, might affect certain strategies on web building? For example, let's say you're in a windier climate or something like that. Like, would that alter the structure or the strategies employed by the spider?
Yeah, it's a very interesting question. Going back to my introduction, talking about the sort of simultaneous physical constraints that this single web structure is trying to meet, I do think it has been found that the spider's webs sort of attains a balanced fitness for each of these goals. So I think, for example, when it comes to transducing prey vibrations, higher tensions on certain radii tend to be more conducive to transducing such signals. However, in environments that are very windy, where the attachment anchor points of the web may be moving at high amplitudes, you may imagine that a slacker web would be more resistant to completely breaking. So it's I can't recall specific studies that directly address that, but I can imagine that the distribution of tension is probably one of the key factors that is being optimized during the web making behavior. And unfortunately, in our current assay, we are purposefully standardizing the conditions of the building perimeter. But hopefully, once we construct our baseline model of the behavior, I think the lab is very interested in exploring, altering the perimeter conditions and the tension exerted on the web to directly answer those questions. Because, and it's an interesting question how much the spider is able to modify the construction rules when it enters a building site based on the measurements that you just mentioned. Yeah.
I actually have a small follow-up. Sorry.
Yeah, go for it.
Small follow-up question to that. I was just wondering, in terms of are there many studies out there that look at the geometry of the webs themselves? Because it really kind of reminds me of an Archimedes spiral, which is really funny because also in for example local search essays of ants and even in flies, for example, one can see these kind of geometric patterns coming up. And I wonder whether maybe the motor programs are kind of similar to the web building motor programs that you can observe.
Yeah, that is interesting. So a lot of existing work actually in the spider literature does focus on that final state of the web, just because it is the most robust structure to actually record. Because you can, once the web is built, you can spray it with sort of metallic substances to increase the reflectance of the silk, which allows you to document, webs you find in the forest, which of course we are not able to do because we don't want to interrupt the behavior. So there is a lot of interesting work quantifying the distribution of geometrical angles and lengths and how those relate to prey distributions and physical properties of the web. So that is very interesting, this type of analyses.
So we have also a couple of questions from the audience. I think we have some time. First one from Flavio Rosses. He says, great talk. Do spiders show plasticity and regulation in their motor patterns while building, for instance, in animals with mutilated or shorter legs?
Yeah, that's a very interesting question. I can't recall the specific sort of labs that did that work, but I do remember seeing studies where exactly the experiment is done that is suggested, removing different legs. And to my knowledge, spiders are robust to some of those manipulations, which goes back to this ability of spiders. It's not a fixed action pattern web making. They're constantly reassessing the integrity of the web, both during construction. Something I didn't mention in my talk is that we actually, despite the stereotype progression of stages I was referring to, we do actually find many spiders in our recordings exit the capture spiral stage and revert to a radii-like stage, appearing to check the integrity of radial lines before then continuing with the capture spiral stage. So there does seem to be a lot of sort of error correcting behavior that may help in such sort of injured conditions.
Great.
I also had a question for Holger, but I can ask it later.
Okay. Well, yeah, maybe you can ask. I just got one more from the audience.
Yeah. No, go ahead.
And then we'll go a bit more general, and you guys can chat. The question was from Snatter. She says, they say, great talk, amazing technique to study the spider's leg movements. My question is a bit about the ecology, though. do you diverse the sample locations too before building the web? Of course, this won't occur in lab conditions, but does this happen in the wild?
Yeah, that is a really interesting question. Just a question of site selection and how many sites are rejected before one is actually chosen. To be honest, I actually don't know. Yeah, it's a very interesting question. One thing to note actually is that in our own data sets, it's very common for a spider, because of their low metabolism, they can actually spend long amounts of time in the protoweb stage, up to sometimes many days before they eventually decide to proceed to the next stages of construction. So I think the spider definitely seems to take its time before committing to the subsequent stages of construction. So it seems very likely that the act of committing to a site is sort of not taken lightly.
Right. Yeah. Interesting. Okay, so that's, I think, all the questions we have in the audience at the moment. I don't know if do any of the panelists have any questions or discussion points? But if Abel, if you did have the question for Holger, maybe kick that.
Yeah, it was a fascinating talk, Holger. I was interested in you talked about sort of our own ears using the sort of geometry of our ears to frequency shift signals depending on their directionality. I was just, me being very unaware of sort of bat anatomy, I was wondering if bats use similar mechanisms. And maybe as it relates to them, because you mentioned they modulate the amplitude of their echolocating calls, but I was wondering if they also modulate the frequencies to track depending on the relative orientation of the prey with respect to their pursuit pattern.
Okay, so yeah, they have also a lot of appendices and ear patterns, nose leaves. So they certainly have frequency patterns that are imprinted onto the echo based on their own shape, and that depends on the direction. So sound from above will have a different frequency pattern than from the front. And they also change the frequencies of the calls quite a lot. So the general pattern is the closer they get to an obstacle, the shorter get the calls and the more broadband in frequency. So to add more color to the picture. Sorry. And that also helps with temporal resolution, because the calls sweep through many different frequencies, and our ear is, also the bat's ear, is processing the sound in different frequency channels. And if you have the same time but cover a broader frequency range, each frequency channel is activated during a shorter time only, and that gives you more precise temporal information, which is again important for the bat to measure the distance to the object. So the time delay between the outgoing call and returning echo is used to calculate the distance to it.
Interesting.
Again, to give it back, there was an amazing talk, incredible data, and I think you can do so many things with that. And so in the discussion, I think you mentioned that the vibration that come from the outside, like wind and so, and that reminded me again of the visual and auditory system that analyze different frequencies. And again, this will probably generate different temporal frequencies, so the vibration might be slower from the wind than compared to prey, and also different spatial frequencies. What is known about how the spiders discriminate vibrations in the net from external sources, wind, compared to a prey item?
Yeah, that is a really interesting topic, and actually spiders have long been sort of a model system for studying mechanoreceptors, just because the slits and cilia are so incredibly sensitive. I read the amazing fact that sort of the energy equivalent of the minimum tactile stimulus that spiders can often tell apart is on the order of magnitude of sort of single photons, just like our own photoreceptors. Yes, so it's very much like vision in its energy sort of limits. So people have studied electrophysiologically the response properties of these slits and cilia and the frequencies that these mechanoreception neurons are tuned to, and they do tend to find that the frequencies, I think, are centered around frequencies that appear to be produced by different types of prey and that are slightly outside the range of a lot of very low frequency environmental vibration. So that's probably one way that they filter out background. But it's an interesting question whether they, I assume they're also able, I assume broadband sort of stimulation from wind on the web tends to, sort of create unidirectional signals, whereas I imagine that prey signals are sort of arriving first and with higher amplitude at particular legs. So I imagine that type of differential sensing is very important in prey localization.
So will you also be able to visualize this with your setup, throwing in prey items or actively stimulating the net with different frequencies, whatever, and see how the spider is responding?
So the work by fellow graduate student Chini Hung in our lab is exactly starting to do those type of experiments. And that's very exciting because a lot of vibration research is done with Doppler vibrometers, and you can sort of sample vibration points on the web. However, the exciting, I think, development in her experiment is the use of sort of high frame rate videography, up to several thousand frames per second, and followed by Fourier analysis, which in theory allows you to have a spatial reconstruction of particular sort of vibration. So she is currently doing experiments in which she introduces prey on the web and then measures the spatial and temporal profile of those vibrations. And one particularly interesting property of this behavior is that the spider will actually crouch and modulate its own posture, which may be playing a role in actually modulating the types of frequencies that it then senses. So it's very much sort of an active sensing behavior, very much sort of like echolocation, I think. It's an interesting similarity.
Yeah. It's more like a comment when you said before. I find this generally fascinating about sensory systems, how tuned they are, how sensitive they are, and what the power of evolution behind that. And that you just mentioned, the eye can detect the photoreceptor, the same for the spiders now in the energy. And for hearing, we say if hearing would be a bit more sensitive, it would respond to the Brownian motion of the air molecules. So evolution drove sensory systems just to the maximum, to the extreme, to get all the information that the animals need.
Exactly, yeah.
So
actually, on that point, sorry, Laura, anyway, because I think that's really like the thing that I find fascinating about both of your work is sort of this idea that perhaps one could think of these behavioral strategies as sort of stimulus response, right? Like kind of inflexible or sort of not cognitive, and yet I think what you both demonstrated and what you sort of have spoken about is that in both cases, in very much like non-human animals, the animals are able to like sort of deploy quite flexible strategies. And I was just wondering, sort of thinking in a very sort of cross-species sense, and how we think about cognition. Maybe this is a bit of an ethereal thing to be thinking about, but like whether we should be looking in the brain for similar sort of encoding strategies to support this kind of flexibility, or whether you think, for example, that because the spiders are waiting for their prey in comparison to bats that are actively sensing and hunting, I mean, maybe that makes a difference in terms of neural encoding. Maybe this is too broad a question, but I was just thinking about cognition and sort of cognitive blocks, building blocks, and how very different behaviors could be supported in these sort of different ways, if that makes sense.
Yeah, I was actually Holger, I think that's a great question, Laura. I'm very interested in the case of bats. Do you think that they are actively representing some running internal estimate of the location of the prey that they then update based on or do you think that the sensory input is continuous enough that they can only rely on reflexive rules? I don't know, because if the prey disappears behind an object that is reflexive of echolocation, then presumably they would need to keep track of where it is going.
Yeah. So there is some sort of internal representation for sure. I have difficulties with the term cognition. I don't really know where it is. I mean, yeah, intuitively it's somewhat clear, but where's the boundary between pure sensory and where's cognition? So, and that can be very dumb. If you have a bat which is hunting in the open air, it can have a very simple search image because there's nothing else except prey usually. There is no leaf flying around, no stones, whatever. So if you throw up a pebble, for example, into an open space where a bat is hunting, the bat will attack this pebble. It'll do this a few times and then stop it. So realizing, oh, there's something strange here. So there's also a lot of familiarity. If they fly through the same cave opening, for example, night after night, they will to some extent rely on familiarity with the spatial memory and just keep going. They still echolocate, so they get some sensory information, but how this is then combined, I think we don't know. If you have animals that live in a very dense habitat, like in a tropical forest, and they drink flower, I think they have a much, much more challenging task than the normal insect hunting bats that I'm usually work with. So they have to identify flowers in a very complex background and navigate in a very complex, dense, three-dimensional space. So we know that they sort of build an internal representation. They keep track of specific objects. So colleagues of mine who do neurophysiology, they find neurons that respond to specific patterns. For example, if, imagine an object just somewhere to the side of you, the bat is flying by. Specific echo properties will change in a predictable manner. The echo will become louder as the bat's closing in. The time difference between the ears will change in a specific way depending on whether the bat is directly heading towards it or passing by. And then neurons that remain active for a specific set of stimulus combinations. So it seems that there are neurons that are able to track an object at a specific position, which we can see as sort of the internal representation of the environment.
Yeah, if I can just add to that question, the question of sort of what is a useful definition of cognition is very dear to my heart. As I mentioned, I came from linguistics, and the way that I like to think about it is in terms of if you put yourself in the shoes of the behavioral algorithm, can you make the decisions that you need to make based on an instantaneous sensory snapshot of the world? And of course, for many behaviors, even flexible behaviors, such reflexive rules may suffice. But language was particularly interesting to me originally and inspiring this interest because, of course, the sort of dynamics of what we are able to say are completely decoupled from the timing and content of events in the sensory environment. So I think a lot of our own complex behaviors are really enabled by the ability of internal states to essentially decouple the complexity of the behavior from the dynamics and complexity of the sensory input at that given moment in time. And so I think it's very exciting to look for model systems that exhibit types of internal states, either based on the time scale that they have or other properties that they have, whether they're discrete or continuous, and to try to find models, in all types of animals that may help inform sort of the circuit elements that we may then go look for in other brains. So I think that's part of why I was excited about the spiders, because of course we have yet to provide evidence, I think, for this being a cognitive behavior. But going back to this question of the sensory landscape that the spider is faced with at any moment in the building behavior, based on our anecdotal exploration of our data sets, I personally struggle to imagine a reflexive strategy that would be able to handle what I consider to be fairly ambiguous sensory inputs that the spider receives. And so I imagine that there may be a role for quite long timescale internal states to sort of remind the spider what stage of construction it is in and what repertoire of behavioral rules it has to execute at that moment in the behavior.
Great. So we have, some more questions from the audience. We have one from long-term friend of the seminar, Alex Kasalnik. He says, do spiders use differences in time of vibration arrival between legs to localize prey? This would require correcting for leg position, wouldn't it?
Yeah, that is a very interesting question. So my recollection of the literature is, yes, that that is very important. The timing difference is very important. And it's an interesting question in what ways the spider has to adjust for its leg position in order to receive that differential signal. That's why I'm excited about Shini's experiments in our lab, looking at the vibration landscape in 2 dimensions rather than just sampled at individual points, because perhaps it will provide insight in how vibrations stemming from a single prey location sort of distribute themselves through the web and may arrive differently at different leg positions. I wish I could give a more precise answer, but yeah, I think those are exactly the right questions to ask. Yeah.
Thanks. And then we just have one final one again from the audience. Again, perhaps can apply to both speakers, but it says, what is the main factor to take into account to separate stereotype behavior from ecologically adaptive behavior and behavioral decisions for either bats or spiders or other animals? That's from Marisol.
Go ahead, Holger.
Yeah, very good, important topic. And I think we often throw in all adaptive behavior just like that and taking the adaptionist approach. Well, I would say, well, we don't know necessarily whether it's adaptive or not, right? We see a bat is doing something on animal. I think also stereotype behavior can be adaptive in that sense. So this, the pattern that we see in bats when they're closing in to any obstacle, making the calls shorter, making more calls, enlarging the bandwidth, is relatively stereotyped, but it has an adaptive function. It's increasing the information, different kinds of information that they need. And we're still not sure yet how flexible it is from, say, repeating the same task once and again, and how much how stereotyped it is. And for example, this very high information update rate, 100, 200 milliseconds prior to prey capture, is still a mystery. Some colleagues have analyzed at what point the bats are still able to change their calling behavior when the prey item is suddenly disappearing. And depending on how close to the planned interception it is, they might change the echolocation behavior. They might even change the flight trajectory and interception behavior. But to some extent, it's also stereotyped. Then they just keep going, emitting those calls, although the prey is not there anymore. And 100 milliseconds before the prey capture, the sensory motor cycle loop is not fast enough to really react to any changes in that. So we don't know why the bat is really doing it. Maybe it's sensory motor consequence. The call emission is coupled to the motor behavior, like the wing movement and the compression of the chest and the exhalation. So I think there's a mix of a stereotype pattern, constraints by all kinds of things, sensory motor constraints, for example, and adaptation, and we cannot completely separate it. That's just hand waving. I have to admit, it's no good answer.
Maybe I can add something for 30 seconds. Yeah, it's a super interesting question. I guess, I don't know if my response is directly related to what you were asking about, but what makes me think of sort of a challenge we faced with our spider, where, as I was mentioning, unlike an animal walking on a surface, the spider's leg positions are modulated by the existence of silk geometry around it. And so we took this approach of finding clusters of stereotype behaviors. But I think thinking about the future of that type of approach, it does seem a shame that we have this, that by clustering, we throw away the contextual information that was maybe adapting the spider's specific movements to the actual goal it was trying to achieve at that moment in time. So I think it's very exciting to think about how these type of clustering approaches can maybe be modified in the future to move away from just sort of simplifying stereotype movements into these clusters and then disregarding the variability within those clusters, and towards an approach that that assigns a stereotyped movement type while explaining the specific variability of that instance of movement from the other executions of that same type of movement. Yeah, that's just a very broad thought, but.
Yeah, that was great. Yeah, thanks so much for all of your brilliant answers and discussion. So that's more or less us out of time now. I'd just love to extend our gratitude again to Abel and Holger for 2 amazing talks. And yeah, thanks a lot again also to the audience for a lot of really nice input. And that just finally would like to plug our next episode, which is actually happening next week on the 19th of April. And we're going to have 2 great talks on navigation and spatial representation in foraging. So yeah, please tune in next week, everyone. Thanks again. And yeah, see you soon. Bye.
Thank you.
Thanks, everyone. Great talk, Holger. Thanks for the invitation.
Bye.
Thanks, everyone.
Bye.